Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosin, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. You can enter your questions for a speaker at any time during the presentation. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Scott Zobbi from Thermo Fisher Scientific.
Thanks. As she mentioned, my name's Scott Zobbi. I'm the Senior Manager for Custom Chromatography Resins at Thermo Fisher Scientific. And today we wanted to talk about some of the advantages of developing a custom resin and how they can be used for purification of complex biotherapeutics. So to kick off, just as you can see on this slide, the landscape of biotherapeutics is changing. So antibodies still represent a large proportion of therapeutics in development right now, but you can see that some of the other sectors, some of the other modalities like proteins, viral vectors, and cell therapies are growing significantly, and they represent a fairly large portion of the therapeutics in development. In addition to that, peptides and nucleic acids are also starting to be pursued for their own unique properties. One of the other things that we see in the market is the size of the molecules are changing. So antibodies being 150 kilodaltons, we're seeing a lot of larger molecules as well, like mRNA, obviously viral particles, plasmids, and things. So with these new modalities and the new complexities that they bring, new tools are really needed to purify them and beyond what their traditional products on the market would allow you to do. And so why would you pursue a custom resin? So there's a lot of different reasons why you might actually look into a custom resin, and so this just actually lists out some of the obvious ones. But there's no suitable resin on the market right now, either affinity or an inert chemistry like ion exchange or HIC. And so you might want to propose a custom resin to do the purification, heavy lifting, and capture through affinity. Also, current resins lack specificity, so you may not have, an inert chemistry like a cation exchanger that's going to allow you to achieve the goals you have. The current process may not be scalable. You may have inherited a process from R&D that uses research use products that really aren't suitable for bioprocess manufacturing. Also, your molecule may be unique. You may have some challenges with that, and you may need to expand your design space in order to create a robust process. And then lastly, you may want to improve the process efficiency and the cost of goods. And that's always a fundamental goal with chromatography and with downstream processing. But when you're trying to create a custom resin, it's even more important. And so within Thermo Fisher, we have a few different routes to get to a bioprocess custom resin. So the route on the left is using our CaptureSelect ligand technology, and the path on the right is using just strictly porous resins. But what we want to do is get you to a bioprocess resin suitable for GMP manufacturing. So regardless of the path you take, whether we develop the ligand and the resin for you under the CaptureSelect path, and you use either an agarose or a porous backbone to develop that resin, or you go down the path of porous and create new novel chemistries, or the clients can show up with their own affinity ligands and we'll do the functionalization for them as well. We really want to get you to that bioprocess product at the end, and that's really the fundamental goal of creating a custom resin. So a few minutes on CaptureSelect. So I want to just talk about that technology a little bit to give you a little bit of background. So CaptureSelect is based on a camelid IG technology, and camelid IGs were discovered over 25 years ago. And so you can see in the upper left-hand corner of the slide, there's a schematic of a camelid IG. And essentially, it's a truncated antibody. So camelids, represented by llamas, alpacas, and single-hump camels, have this truncated antibody form. And what happens within the camelids is that the light chain is lost, and the heavy chain is compressed into a single variable heavy subunit. And that variable heavy subunit, termed a VHH, is really where all the binding happens. So in a traditional antibody, that would be represented by an scFv, but within a camelid, it's a single subunit. And those subunits are fairly small. They're between 12 and 15 kilodaltons, so they're fairly tight and robust. And that actually represents a very good affinity ligand. Because they're small and rugged, they're very good for stability. We can also dial in mild elution and really target very specific molecules as well as epitopes found on that molecule with this technology. Because these are small subunits, we can also manufacture these in yeast, and so it's an entirely animal origin-free production process. So over the years, a number of these products are actually used in commercial scale manufacturing, so they've been completely run through all regulatory agencies. So they're validated from that perspective as well. It's a very tried and true technology. And so on the next slide, we can walk through really the five-step process with which we generate affinity resins, both starting at the ligand phase and then really manufacturing the GMP compliant products at the end. So we actually follow a work package program, and it's divided into 5 sections. And the first section is the library construction. So at the beginning, we'll actually preferentially try a pre-existing library, and we have over 75 different libraries that we've generated over the years. And from there, if we don't, then we'll start, we'll have to actually make a new library, a new target. And from there, the libraries are created, and then we'll go through the screening of those libraries. And so that work package 2 section is probably the most critical one. And so we'll start with a few hundred different potential targets and really try to winnow those down to really about a half a dozen lead candidates. And the types of things that we screen for in that work package 2 are things like specificity for the molecule of interest, binding kinetics, elution characteristics, as well as mild elution, and stability of the ligand itself. And so when you look at all those different parameters, you'll really start to find there's only a few that are really going to be able to achieve all of that. And when you get down to that, you'll take about a half a dozen of those key prototypes, those key ligands, into the next stage, prototype testing. And we'll scale up those 6 targets in yeast manufacturing and actually start producing some prototype resins for screening. And so at that point, we'll actually go in and try and validate the elution characteristics and the stability and the binding characteristics of the plate-based screening, but we'll do it chromatographically. And so from there, you'll really start to sift down again from that about a half a dozen, really down to 1 or 2 lead candidates. And then from there, you'll move into work package 4. And in work package 4, we really identify the lead candidate and then scale that manufacturing up. We start doing bulk yeast fermentation to produce large quantities of material. And then we start also at the same time doing resin optimization. So now you'll start to look at ligand density, really trying to fine tune the elution characteristics as well. You'll look at different backbones, so whether you want to look at Poros or agarose, and that's what happens in that lead selection work package 4 section. And then from there, we move into resin validation. And again, this is done in a 3-lot validation, a minimum 5-liter batch size. We can do batches up to 250 or 300 liters, depending on what the customer needs. But typically, to start, it's a much more modest scale. And from there, once we validate the process, we'll also generate a leakage ELISA and a full regulatory support file, which is suitable for filing with the agencies around the world. And so, really, the goal is again to get to a BioProcess GMP ready resin, and that's really what we're after when we do this. And so if we transition to the next slide, within CaptureSelect, you can see on this slide, this represents the pipeline of our catalog products on the market and in development. This isn't customs. We don't talk about customs broadly. Those are proprietary, so we don't share that information. But within the catalog products, you can see at that top tier, the BioProcess products, those are all the things that are available on the market at scale with regulatory support packages. So they're all suitable for GMP manufacturing. And you can see there's a huge number of therapeutic proteins in there, antibody types, and virus and vaccine resins. So a lot of variety of targets. The next level down is RUO products. So those are research use products. Those are available on the web. They're primarily used for research. Any of those products could actually be converted into a BioProcess product. They just need to be validated. And then below that, stage 1 through 4, those actually represent the different work packages that we saw on the previous slide. And so those are all the things that we're currently working on to move in our pipeline to commercial products as well. But you get a sense from this that there's a very wide variety of different targets you can go after, whether it be, you know, therapeutic proteins, blood proteins, antibody or antibody-like molecules, or viruses and vaccines, or even exosomes. So it's a very flexible technology that really allows you a powerful approach to purifying your product or contaminants away, depending on what you're trying to do. So with that, we'll shift gears a little bit and talk about Poros. So Poros has been around for over 30 years, and essentially it's a polystyrene divinyl benzene resin, so it's plastic. It's rigid, it's a very linear resin, it's very scalable and robust. And so we use that for a number of our inert chemistries like ion exchange, anion-cation hydrophobic interaction. But it also has very large through pores. So it lends itself very well to very large particles, so viral particles, mRNA, plasmids as well. And so you tend to get very beneficial results, particularly for capacity, with those through pores for very large molecules. And then lastly, it's a 50 micron product. So it's all the bioprocess resins that we manufacture are 50 micron in size. And that tends to be smaller than what you find with a lot of bioprocess resins. It's on the smaller end of things. And there's some advantages to that. It really maximizes your resolution, but minimizes your back pressure. So you really are allowed to scale this fully. You can make columns up to 1,000 liters if needed. We have customers doing that now. So it's a very scalable resin, even though it's on the smaller end of the bead size. And so when we develop a custom resin on the Poros side, it's similar to CaptureSelect, but essentially we talk with the customer, the client at the beginning, and find out really what their technical challenges are and what type of molecule they're trying to purify. And so based on that information and whether they have an affinity ligand they've already developed themselves and are bringing to the project, or it's an inert chemistry, we'll go through the process of mapping out what kind of prototypes we want to develop. And the prototype development we'll usually do between 2 and 12 different prototypes for the customers, and then they'll go back and do the evaluation on those. And again, they're looking for things like capacity, separation, removal of contaminants. And then from there, we'll typically do at least another round of prototype development. It doesn't mean you don't get lucky in that first round, but typically to really hone in on a really good lead candidate, it may take additional rounds. And so we'll do at least one round, sometimes two, occasionally more, depending on the customer and depending on the need. But from there, we'll really identify the right formulation. And then from that point, we'll move into work package 2, which is process definition. So at this point, we're looking, can we scale this? And at that point, we'll actually be able to produce material for research use. So you'll actually end up generating about 1 liter to 2 liters of resin at this stage that the customer will receive for research use purposes. And from there, we're really going to map our manufacturing process. We're going to start to write SOPs. We're going to look at the sourcing of the ligand if it's something external. And we're going to look at the formulation of what we're going to manufacture. And then from there, we move into the commercialization, which is very similar to what we saw with CaptureSelect, where we're going to do that validation in a 3-lot system and a minimum of 5-liter batches. And again, those can be larger. It's just that the minimum is 5 liters. From there, we'll also generate stability studies. We'll look at leachables, extractables, and we'll generate a full regulatory support package with CFAs and part numbers for ordering. And so you'll end up with a fully validated, commercially capable bioprocess resin ready for GMP manufacturing. And that's really, again, the fundamental goal of what we do for custom resin. So within Poros, what are the types of things that we evaluate? What are the properties and parameters that we can play with when we're developing those custom resins? So one is the base bead. So we have 5 different base beads we can use within the Poros family. They have different porosities and different pore modes. So depending on the molecule you're trying to target, we can choose the best one for that type of separation. Coating chemistry. So these are polystyrene divinyl benzene. We need to render them hydrophilic. and we have a few different coating chemistries that we use to generate those. Linker length, we can actually play with the length of the linker arm, which the chemistry is at the end of. So depending on, again, the molecule you're trying to purify, something very small might need a very short linker length, something large might need a very long linker length. We can vary those things and really find the optimal one. Coupling chemistry, so how are we actually functionalizing that ligand, whether it be a chemistry or whether it be a protein or something else, to the bead. We have a few different chemistries that we know are scalable and robust. We can play with those and actually evaluate them. Ligand chemistry itself, so when you're doing inert things like cation or anion exchange or hydrophobic interaction, and you're doing different chemistries, you can change and vary those chemistries subtly to drive separations in one direction or another. And the last one, and probably the most critical one, is ligand density. This is always evaluated, and really that's going to drive capacity and resolution characteristics. And when you're generating things with an affinity ligand, it'll also impact the cost as well. And so with that, I want to just share a case study with you where we actually developed a thyroid-stimulating hormone resin with a customer. And so the customer had a request where really what they wanted to do was minimize cross-binding to other gonadotropins besides thyroid-stimulating hormone. And within that family, gonadotropins have a lot of shared homology, so they're very difficult to separate. And so that was one of the key requirements. And also they overall wanted to improve their process efficiency and cost. And so through screening of ligands, and actually it was an existing library that we had, in prototype development, we were able to achieve a commercial resin for this customer that met those needs. And so some of the data is presented here, and you can see on the graph on the left, it shows 3 different prototypes, TSH 3, 6, and 7, and the chromatograms associated with that and their separation. And what they found is for TSH 3, it didn't elute fully, so they got a lot of material in the strip, so it really didn't represent a good target to move forward. And you can see it actually looked very good in the plate-based screenings, but when you started to look at it chromatographically, things didn't work the same. And so that's why you always want to validate these things on column, on resin. So then looking at TSH 6 and 7 on the right-hand graph, what they found is that even though they got better elution with TSH 7, they still didn't get full elution. And so really they chose TSH 6 as their prime candidate to move forward. And from there, we developed the commercial product for them. And what we were able to achieve was 15 mg per mil capacity for that resin. And when they were evaluating this by SEC, they actually achieved, in certain circumstances, 99.94% pure product in a single step. And so you can see it's a very powerful tool, and it can really streamline your process. And at the end, what they found is when they implemented this in their scaled-up process, they were able to improve their recovery from 46% to 71%. So they had over 50% yield improvement in their total process when they implemented this resin. And I want you to think about that for a minute and just think, what would you do if you actually got 50% more product? And what would that impact be? I mean, if you think about it, if you're running 60 cycles in a year, now you only have to run 40, and you're going to generate the same material. And think about the cost savings you would achieve by cutting out 20 additional processes. Think about all the cell culture buffers, all the consumables that go into a process, and all of that is basically money back in your pocket, and just by implementing something like this. And so lastly, I just want to illustrate some of the different programs we've done. So we saw some of the things that we're working on with regards to the CaptureSelect pipeline, but overall we've done programs around oligonucleotides, RNA, DNA, PNA, and aptamers, either functionalizing them or using them as for the target, and that being the molecule of interest. We've also done things around peptides, proteins, enzymes, and hormones, either functionalizing them or those being the target of interest. We've actually seen a growing interest in functionalized enzymes. Viral vectors and viruses for vaccines and gene therapy, carbohydrates and polysaccharides for some very nuanced depletion purposes. We've done functionalization or targets around antibodies, Fabs, and FCFEs. We do a lot of work on ion exchange and hit chemistries, either modifying existing chemistries that Poros has in its portfolio or using new ones, novel ones that customers may need. And then lastly, some dyes and hydrophilic chemistries primarily for some depletion purposes. And so with that, I hope you get a pretty good understanding of the offerings that Thermo has from a custom resin development standpoint, both on the CaptureSelect side for the affinity ligands, as well as the full finished good resins on the Poros side. And with that, I'm happy to take any questions if anybody has any.
Great. Thanks, Scott. So the first question is, where are you manufacturing your custom resins, and which quality systems are in place?
So for the Poros products, they're all manufactured out of our site in Bedford, Massachusetts. And for the agarose-based products, they're manufactured at a third-party OEM manufacturer in the UK. The quality systems are ISO. And so for the Poros systems, it's 13485, which is a medical device standard. And for the agarose-based products and the CaptureSelect ligands, they're ISO 9001.
And what kind of regulatory support is provided?
So for final products that are commercialized, there's full regulatory support packages that are offered. So it's a fairly standard, an industry standard offering. And so you'd basically get a 50, 60-page document that would give you all the regulatory filing information that you would need to use that resin in the GMP process.
Okay. And is it possible to combine multiple targets into one project?
Absolutely. So for CaptureSelect specifically, we can create multi-target libraries. So one of the nice features is that you can basically pool any number of targets into an individual library. And for Poros-specific chemistries, if there were multiple chemistries that you wanted to look at the same time, we'd basically, bundle all that work together. The more things you can do that overlap, the more time savings you can generate, and so there's some cost savings on that in the long run.
Okay. And is there a way to use the identified ligands or custom resins for analytical purposes?
Definitely. So with the CaptureSelect ligands, we can offer those as biotinylated, so those can be used in SPR or SPR-like formats. We can also functionalize them onto Poros analytical columns, so they can be used for titer determination or release assays, depending on your process. For the chemistries customers bring to us, those can be generated as well. For analytical columns, it's a little more challenging to do the biotinylated versions when customers are bringing those in-house, but that's something we could discuss.
Okay. And how much does development of a custom resin cost?
I knew that question was going to come up. So it varies, as you can imagine. So on the Poros side of things, to do the development itself is between $100,000 and $200,000, and that does not include kind of the final purchase of the commercialized resin itself. That's a separate requirement. So just from a development standpoint, it can be a low of $100,000, probably to a high of $200,000. For CaptureSelect, also it varies as well. To do a full target and do the first 4 steps, it's about $250,000 to $450,000, depending on the exclusivity that's required.
Okay. We have just a couple more questions. If we don't get to your question, we'll send those to Scott directly, and he can follow up with you. So the next question is, do your customers always reveal their target?
The answer to that is yes. So the reason for that is, to do the CaptureSelect process to generate a library, you need to know what the target is to actually create that library and do all that screening. And from a BL perspective, biosafety perspective, we need to understand what these targets are as well. So when we're doing the development work, we're safe as well. So we really do need to understand what these targets are. It also helps if they're purchasable. So from release assay standpoint, if you know what the target, obviously if you know what the target is, but it's something that you could actually buy from outside vendors, you know, as a research use product, we'll use that in the release assay test as well.
Okay, and on a similar line, can a customer supply the ligand to Thermo?
Absolutely. So if you have a ligand that you've manufactured yourself, you can supply that to Thermo. We've done that on the Poros side of the business a number of times. We basically set you up as a vendor within our system so that we're able to acquire the ligand from you, and that's definitely a possibility.
Okay. Well, thank you, Chris. Or Scott. Sorry. Thank you, Scott.
No problem.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Thank you very much.